This is our Antimicrobial Lab by
Angelina,
Zara, and me, Darren. Slide 2. Why do some plants and microbes have antimicrobial properties? Plants have antimicrobial defense mechanisms to protect themselves from predators, bacteria, and disease. Microbes produce hormones and chemicals that deter other microorganisms from inhabiting the same space. They do this to decrease competition for resources and nutrients while also trying to keep the population of other microbes down. Slide 3. Description of samples tested.
I picked all of my samples on November 15th of 2021, and I chose my antibiotic samples as basil and cilantro, which were both from my garden, a maple leaf from the tree in my backyard, pine needles from the tree in my front yard, and sienna seeds from a packet that I bought at Meijer in 2019. And my microbial samples were from my house, and I swabbed and plated them on 11/17/2021, and my samples were from the bottom of my garage sink and the lid on my fish tank in my house.
For me, Darren, I chose my antimicrobial samples as an eggshell from my chicken coop back at home, butter from my fridge, an apple skin from the grocery store, tomato skin also from the grocery store, and lastly, a pickle from a fridge. And I did this all on November 16th, 2021. My microbial samples were swabbed on my plate on 11/26/2021 inside the lip of a toilet and the inside of a sink. Slide 4. Description of samples tested. Zara on 11/15/21 picked the following plants from my garden to test my antibiotic experiment. Aloe plant leaves, raspberry leaves, rosemary, chives, and catnip. Zara on 11/16/21 collected the following samples from my antimicrobial experiment. Mold from a car seat, mildew from the bathroom ceiling, and I swabbed the front of my door handle. Slide 5, why we tested these specific samples. For one, it was what we had around the house. We didn't have to go out or buy anything. We worked for the project given. We didn't want to spend money that we didn't have. I ended up having a lot of objects in our house that came from the ground, which was perfect. Was curious to see what around the house had antimicrobial and antibacterial properties. Wondered if the plants chosen had antibacterial properties, and if they were, they are known for being used medicinally. Slide 6, collecting and processing.
For my plant samples, basil and cilantro, they were from my garden, and the maple leaf was from the tree in my backyard. The pine needles were from the tree in my front yard, and I got them on November 15th, 2021. And the sienna flower seeds packet was from Meijer of summer 2019. Then we grinded the samples in a mortar and pestle thing and soaked a paper disc in the grinded material. We used 90 microliters of kanamycin as an antibiotic to help keep the experiment regulated, so it was easy to compare the antibiotic with the microbial. 1 being the plate with no change, 2 being the plate with a negative change, which is tris buffer, and 3 meaning kanamycin was added, so it was positive. On samples 3, I noticed a clearing, as in no bacteria was able to form. On the microbial samples, I swabbed them and plated them on November 17th of 2021, and they were from my bottom of my garage sink and the lid on my fish tank in my house. I grew my bacteria at room temperature until November 21st, 2021, and placed them on the fridge on this same day on the potato agar plates. Came to lab on November 23rd, 2021, then grabbed an agar plate and inoculated the plates with the fish tank bacteria and the sink bacteria, and also labeled those 1, 2, and 3.
Slide 7, collection and processing by Zara. Plant sample collection. I collected my plant samples on 11/15/21 by going to the garden with sandwich bags, and I put the 5 plant samples in their own bag. I then labeled the bags with the name of the plant and the date collected with my name on them. Plant sample processing. I brought the plant samples in the lab the following day on 11/16/21. And I ground up leaves individually by using a mortar and pestle. I put a few drops of tris buffer into the bowl with the ground up plants to make a paste. I used a small paper disc to soak up the solution. I then took 2 agar plates and divided into 8 sections on each plate. I put bacteria on the plates and spread it all over. One plate I put a plus sign and a negative sign on 2 sections. These were my positive, kanamycin, and the negative, tris buffer control. I put my plant names on 3 sections per sample. I used paper disc to soak up the kanamycin and placed the plus section on it. I used the other paper disc to soak up the tris buffer and place it in the negative section. I then took my paper disc with the soaked up plant solution and then put them in their labeled sections, using 2 plates total. They sat for 1 week until analyzing them in the next lab. Slide 8. Collecting and processing SARA. Microbial sample collection. I collected microbial samples on 11/16. I brought 2 agar plates home to put the samples on. I first grabbed a new chopstick out of a package and scraped some mold from the various places in my car seat with it and put them on the plate labeled car seat with the date and my name. I then grabbed the other plate that was labeled half with bathroom ceiling and labeled the other half front door handle. I grabbed a Q-tip and rubbed off some mildew from the bathroom walls and the ceiling, and I put it in its labeled half. I did the same thing with the new con swab with the door handle and put it in its labeled side. I then left it on my computer for a couple of days and let it sit in the fridge until the next lab. Microbial sample processing. I brought my 2 lab plates on 11/23 and took 2 new agar plates and separated them into 8 sections each. Each plate had control sections with kanamycin with a plus, tris buffer with a negative, and there were 6 other sections, 3 from each location, making a total of 2 microbial samples per plate. I put the bacteria all over the plates. Put my controls on each plate, then I used the inoculation loop to put microbial samples on their labeled sections on the new plates from the original potato agar plates. The samples I put on the new plates were from the same place, but from different areas that grew on the plate. This is collection and processing by Darren. So for my plant samples, I got an apple and tomato from the store. And lastly, I had my pickle in my fridge. And these were all picked on November 16th, 2021. Then I grinded the samples in a mortar and pestle and soaked the paper disc in the grinded material and placed it on the plate. We used 90 microliters of kanamycin as an antibiotic to keep the experiment regulated. And this makes it easier to compare the antibiotic with the microbial. These are listed on my plates as the name is the objects I used. For example, I said apple with a negative sign, meaning I used tris buffer, and I used a plus sign, which meant I used kanamycin. And for my regular variable, I just left it as its original name. Slide 10, results.
This is our first slide of results. You can see in the pictures that contain the plus, which means they contain kanamycin, that they have a clearing, which means they contained antimicrobial properties, meaning no bacteria could grow right there. And you can see it in all 6 of these pictures right here. And then also, fish tank in the sink on slide 3 contained antimicrobial properties as well. In Zara's results, you can see the chives saw multiple clearings. You can see 3 clearings right there, and that means they had antimicrobial properties. Same with this rosemary. And this car seat mold right here also contained antimicrobial properties.
Steps we should have taken to continue this experiment. 1, we should repeat the experiment with the bacteria showed in the antimicrobial properties. We should repeat the experiment with the plants that showed antibiotic properties. We should give more room for each sample so the results are more clear. When inoculating bacteria onto a new plate, we should take each sample from the same spot on the potato agar plate and consider each new spot on the new sample. Conclusion. The samples that contained 90 microliters of kanamycin had clearing. Samples that contained tris buffer did not have clearing. Fish tank 3 and sink 3 had clearing and showed antibiotic properties. The chive samples had clearing. Aloe samples had small speckled clearings. The wall had a slight brown. 1 to 2 had clearings. Car seat 2 had clearings. Door handle 3 has a slight clearing.